Hi, it's Jared Smith, and in this podcast we'll cover the fundamental principles of the cell cycle control system. So we're going to start with a broad overview of the cell cycle in general. From there, I'll discuss various checkpoints such as the G1, G2, and M checkpoints specifically. We'll look at regulatory molecules that run the checkpoint such as cyclins and cyclin-dependent kinases, and the stop and go signals that these regulatory molecules are going to give. I'll specifically mention internal signals such as MPF and external signals such as PDGF, and finally we'll talk about disruptions in the cell cycle control system as in the case of cancer. So we're going to start here with the cell cycle. In general, it's a process that results in the creation of two cells from one by cell division. If you look at this image here, you see the different phases of the cell cycle and about how long each phase lasts. So this purple ring represents interphase, which consumes about 90% of the cycle. Interphase is going to be broken down into three phases: G1, S, and G2. So I color-coded these so you can differentiate between now. G1 phase, G is going to stand for growth. So as we see here, this small cell is going to be made larger, and it's going to grow. So after that, the cell is going to enter into the S phase. It's going to be represented by this pink section here. The S is going to stand for synthesis, and as we see here, the one chromosome is changing into two. It's duplicating, synthesizing. So finally, it's going to enter into the last stage, the G2 stage in interphase. And as we see here, the G stands for grow. So the cell is just once again enlarging. So the cell enlarges, duplicates, and enlarges, or synthesizes here. And then interphase is going to be done. So then we're going to move into the M phase. That's this small section here that's going to take about 10% of the entire cycle. M phase here, it's a thorough process, but it involves mitosis, as is the separation of the nucleus, and cytokinesis, the separation of the cytoplasm, and thus the entire cell. So I'm going to dumb this down into a simple process in which it is actually a thorough one, but here's a parent cell you see. It's going to divide and create two daughter cells. You see these daughter cells are the exact same size, the exact same chromosomes. They're going to be identical to the parent cell. So mitosis is going to change a parent cell to two daughter cells that are identical. So that would be the cell cycle in general. Now we're going to move on to the checkpoints. So checkpoints, they run the cell cycle basically and tell it when to enter into the next stage, whether it be G1, S, G2, or M. These checkpoints can be looked at as stop or go signs. So here's a stop, here's a go. If a cell is acceptable, then the checkpoint will allow it to pass, as represented by the go sign. If it's not acceptable, it will be rejected, as represented by the stop sign. Oftentimes, if a cell is not allowed to pass through a checkpoint, it will enter the G0 stage, where it will no longer divide. Many cells in your body are actually in the G0 stage, such as muscle cells. Specific checkpoints can be seen at the G1 phase, G2 phase, and M phase. The G1 checkpoint seems to hold the most significance, as cells that pass through it will most likely complete the entire cycle. So we've got here the G1 phase, the G2 phase, and the M phase. So G1, if it doesn't get through G1, it's going to go into the G0 phase, as I just stated, but if it does, as I also just stated, it's probably going to complete the whole cycle. So, however, if the cell goes all the way around and makes it to the G2 checkpoint here, it still has the chance of having some minor deficiencies where it can still enter into the G0 stage. But this G2 checkpoint, if it goes through it, it will make it to the M checkpoint. And if it goes through that, it will divide into two cells, and those two cells will often further divide into two more cells, and the process continues, or they'll often just turn into a body cell and not divide anymore. So G1, this teal color here, G2, red color here, G3, or M, sorry, and from there it will complete the cell cycle. So now we're going to talk about specific stop and go signals. So what stop and go signals regulate these? Kinases are what we call them. The kinases are usually in inactive form. They're just surrounding the cell, and they're not really useful. So I've got this visual here. I represent the kinases with the green and the cyclins with purple. Now, cyclins are going to be molecules that are constantly fluctuating around the cell. So the kinases are cyclin-dependent kinases. So they're going to depend upon these cyclins to actually move them. So they're going to go from inactive to active by connecting onto them. So in this animation, we see that they're connecting onto them. So we go from this to this. And now they can control the checkpoints and allow the cells to pass through. So we're going to go even more in depth with examination of an internal and an external signal. So this internal signal is going to be MPF that we're going to talk about. So here's the visual of the cell cycle. If we remember, this green was the CDK, or cyclin-dependent kinase, and this purple was the cyclin. So in MPF, these two are going to come together, as I just stated with the CDK, and they're going to form MPF. And the MPF is going to stand for M-phase promoting factor, basically. So it's going to allow a cell to pass through the G2 checkpoint and into the M phase. So it's going to go around here. We see it's coming around. It's aiding the cell and passing through. And finally, it's finished its job. So what's it going to do? The cyclin's going to come off, and the CDK is going to continue on. But the cyclin is going to come around here and be used again, and the CDK is going to continue around the cell cycle until it comes to that. And it's going to keep repeating. So now we're going to talk about an external signal, such as PDGF. And in an experiment done long ago, we see two vessels here. One vessel has cells, and the other one has cells. But the difference is this one has PDGF. So what do we see? We see more cells in this one. Why is that? Because PDGF stands for platelet-derived growth factor. So it's a growth factor, meaning it's going to aid cell division. So this is going to be an external signal from outside, another substance. And in this first vessel, we see the cells aren't dividing. In this second vessel with PDGF, they are dividing. So it's going to help with cell division, and it's going to be a signal. So now we're going to talk about cancer. So cancer is a serious problem around the world, and it's going to start with one cell. Now this one cell, it can happen to it. It can go through transformation, and this transformation will turn it into a cancer cell. So now the cancer cell will oftentimes accumulate and form a tumor. So the tumor can be benign or malignant. And the benign tumors, it's just affecting one organ. So benign, it's not going to be that harmful. It can be removed by surgery. What gets complicated is when it turns malignant. So in malignant tumors, it's going to, through metastasis, spread to other organs. So now multiple organs are affected by it, and this is where the serious cancer comes in, where people often die from it. So cancer, it results from disruptions in which a cell transforms into a cancer cell, and that would be the cell cycle control system, and I hope that's helpful.